Thank you. Yeah, it's great to be back here at the LSR. I remember so many talks in this very room and happy lunches and so on. So yeah, really great to be back after so long. I think it's about 20 years since I started here. The work I'm going to tell you about today was largely funded by the Leverhulme Trust via a Research Leadership Award, and here are the names of the people involved. My aim for today, I want to give a bit of background on stereopsis. I gather many of you are not working on stereopsis, so I don't want to, dive straight in without discussing that a little bit. I'll tell you about some of the behavioural experiments we've done on praying mantis, and then if I have time, a bit on the physiology, although I'm totally prepared to skip that if we look like short of time. And obviously we're a small group, so definitely, don't be afraid to ask questions or stop me or anything. Much more fun. Okay, so what is stereopsis? Well, it's a word that in Greek means solid sight, and the Wikipedia definition talks about it meaning the perception of depth and 3D structure. Stereoscopic vision is just a synonym for stereopsis as far as I'm concerned. I prefer, for obvious reasons, given that I'm talking about insects, to avoid things like perception and just go on information about the 3D structure of the world that's obtained with different lines of sight to the same point in space. And it's that last bit that's critical for me in defining stereopsis. So the basic geometry is just triangulation. Okay, so if you're looking at a scene like this with your two eyes, and you can somehow extract the angle alpha that's subtended by your interocular baseline at the flower and recognise that that angle is larger than the angle beta subtended by the bird, or the bird subtends at your interocular baseline, then you know the flower must be closer than the bird. And that's the basic geometry behind stereopsis. I thought, since most of you do not study stereopsis, it's always helpful to begin with a bit of, a why should you care section. I think stereopsis is important partly clinically, so as Bharat mentioned, I am interested in the clinical relevance of stereopsis. It seems that human stereopsis is particularly challenging. It requires good vision in both eyes individually, good ocular motor control to point both eyes at the same point in space, and the relevant cortical mechanisms to put the result together and infer depth. And there's wide areas of human visual cortex that's involved in this process. And it seems that it's something that can quite easily go wrong during development. So you have situations like strabismus or squint, where the eyes end up pointing in different directions, amblyopia or lazy eye, where one eye ends up visually impaired just because the brain isn't listening to the signals from that eye rather than because of any problem with the retinal image. In older people, you may get diplopia or double vision. And actually, these binocular disorders are the most common cause of visual impairment in children in the developed world, where thankfully, of course, we've eliminated many other causes of visual impairment. So if you can understand how stereo vision works normally, you can start to understand what may go wrong in these conditions. And some of my clinical work has been developing a test to measure stereo acuity, so the ability of stereo vision in children in the form of a game on a 3D tablet that's more fun and engaging than conventional stereo tests. Nowadays, we live in exciting times for people working on stereopsis because there are so many 3D displays like that 3D tablet. And so again, people need to understand, well, how do we perceive depth in a display like that? And this could be an entertainment, watching a football match, but it could also be in really serious applications like a surgeon doing laparoscopic surgery, looking inside the patient via a camera on a 3D display. So, any compressions or distortions of depth could potentially be really important there. Another angle is machine vision. So we can now create algorithms that can see stereoscopically and potentially use them in autonomous vehicles and other robotic applications. And I actually think insect vision is quite an exciting area in this context. One of the things I always think is striking is just how late science was to discover the existence of stereopsis. And I think it's particularly puzzling since vision, and even binocular vision, has occupied the finest minds of humanity since forever. So Ptolemy, in his optics, is discussing why do we have two eyes, and he understands that it helps us see better in some way. People also understood that specifically depth perception was impaired with one eye. So this is a beautiful engraving by Rubens for a book on optics by Agrolonius, and they're showing an old man who's only got one eye, and he's making reaching errors. So he's trying to reach out to this stick held by the putti, but he's making errors because of his binocularity. Try and get my laser pointed out. People assumed this was due to vergence. So here's an engraving from Descartes' book on optics, and he's showing a blind man inferring the distance to objects by how far he has to point two sticks together to touch them. Presumably he's driven to such lengths because his guide dog looks a bit rubbish. So all these people thought that binocular vision must help depth perception because we could sense the divergence angle, right? So you can tell when something's near versus when it's far away. But ironically, humans are terrible at sensing our own vergence, and we get very little information from that vergence cue. And the consequence is that while human stereopsis is very good at detecting relative distances, such as this purple object is nearer than the orange one, it's very bad at detecting how far away any of them are in absolute terms, in meters, which is intriguing. So human stereo depth perception is driven very much by relative disparity rather than vergence. And actually, this is a slide that I had years ago from Fred Miles and have been using it ever since. It's a great slide, thank you, Fred. So there's somebody fixating a distant object like a mountain, so it's projecting to the fovea in both eyes by definition. And then this nearby tree, you'll notice it's on the right of the mountain as seen from the left eye, and the left of the mountain as seen in the right eye. So it appears on the retina with a disparity, a shift between them. And that disparity I'm going to be talking a lot about, that's the cue to stereopsis. So Leonardo da Vinci intriguingly got really close to recognising the importance of this disparity cue. He recognised that, ignoring vergence, one of the issues is that the two eyes are seeing different retinal images. And he talks about occlusions in particular. So he's saying, if you've got an object like this sphere, it's going to occlude different regions of the background as seen from the left eye versus from the right eye. And he points out, this is why my pictures never look 3D, right? I can get everything else completely perfect using this newly invented science of perspective, but I just can't get that disparity information. And it's really frustrating to me that he didn't go that little bit further and think about, well, so maybe I could draw two pictures, one for each eye, and use an arrangement of mirrors to invent the stereoscope 200 years before. Wheatstone, sorry, more than 200 years before Wheatstone, finally gets to it in 1838. And he does do exactly that, draw images rendered for the left and right eyes, use mirrors to present them, and finally shows that this alone suffices to give a really strong perception of depth. So this is the reason why I use this example of the flower and the bird, by the way, because as discussed, human stereopsis can't tell how far away either object is. So stereoscopic viewing, contrary to how it's often presented in kind of elementary textbooks, is actually no good for what I want to call range finding, sort of distance. That raises the question of what it is good for, which I'm going to return to. But first, I also want to mention another failure of science, and that was to realise just how widespread stereopsis was. I think because it took us a long time to discover stereo, we kind of assumed it must have taken evolution a long time to discover stereopsis, and therefore stereopsis only arose as a final flowering in man and a few of the higher animals. So there's this view that you still find again in textbooks that stereopsis is limited to, say, predatory mammals with front-facing eyes, like monkeys and cats and so on, also drawn on the machines that we create. But actually we now know that you also find stereopsis in lateral-eyed mammals, including mice, including prey animals like horse and sheep. You find it in predatory birds, for sure. You find it in amphibians, like the toad. Even recently discovered in cephalopods, the cuttlefish, and of course in insects, where it's really been demonstrated in the praying mantis. So that's what I'm going to talk about mainly. You might reasonably ask, well, what about other animals? What about jumping spiders or dragonflies or whatever? I think it's very possible that stereopsis may be extremely widespread, especially, by the way, since the previous slide implies that stereopsis has evolved independently at least 5 times, because even the common ancestor of birds and mammals has a different substrate for binocular vision. So it's been discovered a lot of times in evolution, which suggests to me that it's very useful. But it's very difficult to do an experiment to prove that an animal has stereopsis, so mainly we just don't know. One little detail I thought I'd mention, it's often stated that front-facing eyes are an adaptation to stereopsis. But I think that's a bit of a fallacy. What front-facing eyes give you is a wide binocular overlap. It's not clear that's useful for stereopsis. It's certainly not exploited in humans, where we have a binocular overlap of about 70 degrees, but stereopsis operates only in about the central 20 degrees. Right? So we're not exploiting that. What I think is a more obvious evolutionary specialisation for stereopsis would be a wide intraocular separation, because that really improves the precision and the range of stereopsis, given your visual acuity. So on that basis, I think we should be looking at animals like the hammerhead shark, which does appear to have evolved a very wide intraocular baseline and has a substantial binocular overlap, and the stalk-eyed fly, which could potentially have stereoscopic vision not only in front of it, but also behind. But let's turn to an insect where we know much more about its stereopsis, and that's the praying mantis. Now, you might be wondering, how can you tell whether an insect has stereopsis? And the answer is in a beautiful series of papers by Samuel Russell from the 80s, starting with this one in Nature. So praying mantids are visual ambush predators, so they spend a lot of time sort of lurking, and if something comes within range, they'll strike out their spiky forearms and grab it. So you can see it's rather critical that they know how far away the animal is when they launch their ballistic strike. It's very rapid, it's just about 40 milliseconds. People have long speculated, again since the 19th century, that they were using binocular disparity, so triangulating on this angle delta, but Russell proved it by the simple expedient of putting base-out prisms in front of the animal. So of course prisms bend the light rays, but you can't expect the mantis to take that into account, and so it's going to make errors in its catching. And Russell showed that indeed the pattern of strike errors, as he varied the power of the prisms, varied as predicted, and that's really powerful evidence that the mantis is using binocular disparity to judge distance. And Russell was kind enough to share with me this video of one of his experiments. The mantis is upside down, that's how they like to be. They hang out in their cages upside down, and he's tantalising it with this prey-like object. And you can see that viewing it through the prisms, it's striking, even though it can't actually reach the prey. So, unlike humans, it seems that mantis stereovision does do range finding. And at this point, I want to talk a little bit about the different visual systems that make that possible. And for this group, which I don't think anyone here works on insects, I thought it might be useful to include some background on insects and insect vision. So a question I'm often asked is, what about those compound eyes, right? Doesn't that make everything different? Well, yes and no. So there's this long-lived myth about insect vision, stemming from what you see if you take off the insect cornea with all those little, that array, those facets of lenses, and you look through it, you see many copies of the world, right? Like many copies of this number 7. This does not mean that this is how the insect sees. And scientists have long appreciated that, but in popular culture, there we go, there's the idea, I'm sorry the sound is indeed not playing, but this is from a 1958 documentary, The Fly, about a scientist who suffers an unfortunate lab accident and transplants the head of a fly onto his own. This is an unfortunate wife discovering the fact, and the film shows us seen through the scientist's fly eyes, seeing many copies of his wife. This is not, surprisingly, how insects actually see. You can still buy all these educational toys that, purport to show you how a bug would see, but it's kind of, if you think about it, how would that even work, right? How would a bee land on a flower if it was seeing many copies of the flower? So what actually goes on is this. Simple eyes like humans, we have a wide pupil, and so we collect light across a wide, area of the pupil, and then we have to focus that down so that all the rays from a given direction land on a given photoreceptor. And of course, we do that by the accommodative power of our lens and also the power of our cornea. And then rays from a different direction are again collected all across the pupil, but focused down to a different photoreceptor, making an image. In the case of compound eyes, most of the light is wasted, it just bounces off the eye. Each photoreceptor's got its own individual lens, and so the photoreceptor that collects light from this direction only collects a small fraction of the light into the eye. And similarly, light from a different direction gets detected by a different photoreceptor. So this makes insect eyes very inefficient, and they're also very low resolution, but the end result is just a retinal image, same as it is in us. So for me, the big difference between insect and human vision, and stereopsis in particular, isn't really that insect eyes are compound, so much as the fact they're fixed on the head. So this problem of vergence that we saw earlier doesn't apply to insects, you could just assume it's no. Small footnote, because there's some really interesting work just coming out, the paper hasn't been published, showing apparently vergence movements through movements of the retina little muscle movements shifting the retina under the array of lenses, which is fascinating. So I just want to acknowledge that. But to a good approximation, insect eyes are fixed, and therefore you don't need to worry about versions. And so the mapping from disparity on the retina to position in space is fixed and can be assumed to be known. And I think that's why they can do range finding, whereas we can't. So this is a talk mainly meant to be about insect vision, but I think it's useful, given that you guys mainly work on primates, to say, well, if human stereopsis doesn't do range finding, what is it there for? Well, of course, even revealing which object is closer, it can be very, very useful, as anyone will tell you who's tried to thread a needle with one eye closed. Really hard, right? And when you've got stereopsis, you can tell when the needle and the thread are lined up. So that's useful in itself. You can reveal depth structure across the visual field. So like imagine the eyes looking at the scene here. This is a machine stereo algorithm which has used the small disparities between the images to get the depth structure. Even if you don't know how many meters away the red and the blue is, it's still very useful to see the boundaries. It can help you interpret the scene, see where the edges of objects are, and so on. One interesting application is in locomotions and walking over complex terrains. And I think this work from Kate Barnon and collaborators caught my attention because this is another area where I've noticed stereopsis in my own life. As a stereo researcher, I sort of play around and like close one eye, you know, things get better or worse. In most of my life, it doesn't really make any difference, but you try fell walking or fell running, it really makes a difference there. I could believe that. And that also is suggestive, given that stereopsis has been found in the horse. You know, what do? They're not threading needles, they're not catching prey, but they are locomoting at speed over uneven terrain. So maybe their stereopsis is helping them out with that. But there's something else stereopsis can do, or primate stereopsis can do, and that's called camouflage breaking. So it can reveal depth structures that aren't visible monocularly. And here's an example of what I mean. So this is a pattern of random dots, little square dots in the background. But did you see a disk appear briefly just there? So defined by the shift relative to these two frames. And if I present frame 1 to the left eye and frame 2 to the right eye, then you can extract depth information from that. And that was pointed out by the Hungarian psychologist Beli Ules, hugely influential in the field, and he suggested that this might be why stereopsis evolved in primates, to help us pick camouflaged beetles off bark, you know. It's been an important use of stereoscopy historically, for sure. So you can, in aerial reconnaissance, you can take photographs, and if you take them as you fly over, you effectively have a very large baseline, and then you can view the successive photographs in a stereoscope, and a human observer, of course, long before there were algorithms on computers, could extract the depth from that. And I like this quote from a book written after the First World War and the experience of aerial reconnaissance, saying how bridges, observation towers jump into view in the stereoscope when often they've entirely eluded notice in the ordinary flat picture. So this camouflage breaking has been useful in a human arms race and possibly in an evolutionary arms race as well. So how do you achieve this camouflage break? One of the problems with complex natural scenes is that they often contain many similar elements. So like two identical apples, let's say, or the chairs where every single chair has an identical corner as I look around this room. And you have to successfully match up which apple belongs to which other apple in the other eye, if you see what I mean. Otherwise, if you make the wrong pairing, you're going to infer something completely different about the depth structure. So figuring out which little bit of a scene belongs with matching bit in the other eye is referred to as the stereo correspondence problem. It's a huge deal. Correspondence problem in general is a huge deal in computer vision because it's also important for motion and optic flow and so on. Originally, psychologists thought of the stereo correspondence problem in random dot patterns like this as like, super complex. You have to figure out which little dot in the left eye matches which one of the hundreds of other little white dots in the right eye. But over time, we realised that that's really the wrong way of thinking about it. We should be thinking about something much simpler, which is where is the binocular cross-correlation highest? So you've got to imagine taking little patches of the left image and sort of sliding them across the right. This is what a correlation function does, until you find the bit where they match best, so where the correlation is 1. So in most of the image, it's that shift, and then for this central disk bit, you need a slightly larger shift. And so that's a good way for an algorithm to detect the differing disparity in those regions. So this seems to be how human stereopsis works. We cross-correlate little local patches of the left to right images. Those patches seem to correspond to the receptive fields of neurons in 5-visual visual cortex, and the inferred disparity that presumably your brain then extracts from it is where what shift you have to apply in order to get the peak cross-correlation. So, backing that up is the idea that inverting correlation, messing with this cross-correlation, really destroys human depth perception. So, I'm afraid I haven't brought any 3D glasses, and it would really only be for this slide. But on the left, we have what's called a correlated stereogram. So it's like the one we saw before, where there's a pattern of black and white dots. The centre region has been shifted. If you put on 3D glasses, you see it standing out in depth. On the right, we have an anti-correlated pattern. So now the white dots in the left eye have been replaced with black dots in the right eye. So the correlation is never going to be 1, it's going to be -1. And this just looks a complete mess for humans. You don't even see any depth structure at all. Okay, so that's great. Seems to be a really good description of humans. Bruce did some really influential work with this in monkeys. People have also studied it in other vertebrate species, and it all seems to work just fine. And it's the go-to method for machine stereo algorithms. They're all basically built on cross-correlations. So a few years ago, I was thinking about this and was struck by these similarities. So, as I say, people haven't really studied other animals in detail, but humans or birds or machine algorithms that people have come up with, they all basically do stereopsis in the same way. And so that raised, and that same way being that they solve the correspondence problem using cross-correlation, which means they can break camouflage, as you say, the dot pattern, and get this lovely depth structure across the visual field, which may be why their stereo vision evolved. So it then seemed to be really important to study some of the other animals, and especially really simple ones like insects, because as I see it, there are basically 2 possible outcomes, right? Either insects also do stereopsis the same way, in which case, given the vast differences between these 2 systems, that strongly suggests this is the only good way to do stereopsis. Don't bother investigating other ways. Or you may find that they're doing it in a different way, and that could give you new ideas for machine stereo algorithms, for example. So that's what really motivated me to go after stereopsis in insects, abstruse as this problem might seem in itself. However, the first problem we faced was a bit of a practical one, which was like, how do you create a 3D display for an insect? So Russell had come up with these prisms, which are great, but essentially they're just pushing everything nearer or further. And we wanted to be able to display complicated images like these random dot patterns. So Vivek Nityananda, who was at that time a postdoc in my lab, is now a colleague of mine at Newcastle University. It took a long time. We figured out an alternative approach eventually that we could demonstrate worked.
Can I ask you a question?
Please, yeah.
So when the praying mantis is doing its strike, are they adjusting where they strike based upon the depth? Or are they waiting for the fly to enter basically a particular zone that they always sort of stereotypically strike at?
Basically the latter. Okay. So there's a nice paper from Russell, I mean it's n=1, but he's showing a particular mantis and demonstrating that its strike trajectory does change somewhat, depending on the distance. But basically they have a fairly narrow catch zone. In our species, it's about 2.5 centimetres in front of them. And so if something comes within range of that, they'll launch out and grab it. And they have fairly little scope to go nearer or further. We try to look at that ourselves. There's a lot of strike-to-strike variability. So it's a little difficult trying to disentangle, are they systematically reaching nearer for the nearer objects? So yeah, basically we think of a catch zone. So this is a video that I wanted to sound on, but I'll just sort of talk over it. Because it just shows some nice footage of Vivek applying 3D glasses to a praying mantis. So there's the mantis in its box, and what he's going to do is cut out little 3D glasses from spectral filters. So we're using different colours to separate the images for left and right eyes. He's anaesthetised the mantis by putting it in the freezer briefly. He's explaining that. He's then immobilised it with plasticine, our sophisticated restraint technique. And now he's cutting out the filters, and he's going into this teardrop shape, and he's going to use beeswax and resin as a sort of natural glue that isn't going to damage the animal. He's melting it on a melter, and he's just going to dab the filters in place so that they're held by that dab of beeswax. And then he's also going to put on the thorax of the animal a holder that's going to hold it in place during the experiment. And none of this seems to bother the animal. It goes back to its home cage and just sort of behaves as excitingly as mantids generally do. So that seems fine. And then for the experiment, we're putting it upside down because that seems to be where mantids are most comfortable. Actually on the same stand that Russell used in his experiment, which he very kindly donated to us. And now we're testing the 3D glasses by arranging a situation where the mantis is far out of the catch range from the screen. So the screen's 10 centimetres away, there's no way the mantis can reach that. And so it wouldn't usually strike at objects on the screen. But when we present objects with disparity, you're going to see that the animal reaches out and strikes. I always think of it a bit like little kids. You sometimes see them in 3D movies trying to grab things that appear in front of the screen. There you go. So it did take us a long time to figure out a suitable set of 3D glasses that would achieve that, but we eventually did. And so the key experiment demonstrating that was to compare what we call the crossed disparity condition, where we set up a situation where the left and right eyes images were arranged so as to place a virtual object in the catch range, and then a control, this is obviously crucial, where you swap over the left and right eyes. So if it just liked seeing, two objects going around a screen, it should strike at the uncrossed condition. But in fact, it very rarely does, maybe never's a bit strong, but it hardly ever strikes in that condition because the lines of sight diverge, right? So we wouldn't expect it to perceive anything within range. So that's great, but all it's done really is replicate Russell with a different display technology, which we had to do first to prove that our technology worked, and now we can do the interesting stuff, probing insect 3D vision. Then we have another problem. We wanted to go after the first question, like, do they use binocular cross-correlation? Cells, monkeys and everything. The problem is they only strike at things they think are prey, they only eat live prey, they only strike therefore at things that move. It's interesting, sometimes you see, if when you watch them hunt, if prey freezes, they sort of know it's there and they're still attending to it, and you can see them sort of doing peering movements sometimes, but basically, I never see them strike unless the prey's unwise enough to make some little move, maybe a twitch of an antenna, and then they go for it. So we have to have moving stimuli. We can't do the classic human psychophysics situation. So we made a random dot pattern with a moving simulated bug, and this swirling stimulus that you saw earlier, the motivation is just that empirically we just found that seems to get them going. It goes round and round and round, it gets them all excited, and you can see their little heads going around watching it, and then it comes to rest, jittering just in front of them, and then they reach out and grab it. So that's the reason for this swirling pattern. And we replicated that with a random dot pattern. So we take a chunk of the random dot pattern and we have it swirl around, shown in slow motion here. And again, we've got a patch with the left eye and a patch with the right eye, so we can vary the disparity. And again, we can use the uncrossed geometry also in this random dot pattern. So when we did that, we had no idea what we'd find or whether mantis stereo could even operate with such highly unnatural stimuli. Generally, people had probed their strike behaviour with black objects on a white background, which kind of simulate a bug against the sky, I guess. So that's much more naturalistic. Whereas we were using a stimulus like this, using round and round and round. So it's actually camouflaged perfectly on any frame, although the motion, of course, breaks the camouflage. So here are results from that experiment. So what I'm plotting here is the strike probability, so proportion of trials on which the mantis struck, for the uncrossed initially on the left, just when you have a normal stimulus on the screen, and for this cross disparity situation. And you can see there's a very statistically significant difference. They're much more likely to strike when the prey is simulated at the correct distance, even in these really complex random dot images. So that's cool because it shows that their stereopsis still works in these images.
Do they start striking right from the first trial, or do they need some time to get used to the?
I don't think we've really analysed that, but sort of just from what you see, basically from the first trial. So actually, their motivation varies a lot. So there are some days and some mantids when you put them on the stand and they don't strike at anything the entire session. And that's not very helpful, obviously. And then other times when they're more motivated and they strike more. And the females tend to strike more than the males, probably because they're bigger and they need to hunt more. So what we do in our experiments where we're using wacky, weird stimuli like these random dot patterns, which elicit quite low strike rates, presumably because they're so unnatural. We start off with the swirling disc stimulus, which is our sort of optimum one. If they strike at that, we go, okay, they're motivated to hunt, and then we run a series of experiments with the dot patterns. And we do basically one trial every 2 minutes, so they, aren't getting sort of zapped over and over again and have a little time to recover. But they don't really seem to habituate or get discouraged by the fact they're obviously not catching anything. And then at the end of the experiment, we then run the black swirling disc again. And if they strike at that, we go, great, their motivation was high throughout this entire block, we'll use it. If they don't strike at the beginning, we don't even bother. If they strike at the beginning but not the end, we throw that data away because we don't know at what point they became unmotivated. Even though we're randomly interleaving, it's just, and that doesn't happen very often, luckily. So it's basically their motivation fluctuates for unknown reasons. It doesn't seem to be correlated with hunger. So we just try and use them when they're in the mood to hunt. Good question. Great, so now we wanted to go on to the mixing up the correlation, okay? So we could do that in exactly the same way as with the previous random dot pattern. We did it for anti-correlated and also uncorrelated, where we've just generated a completely different pattern in left and right eyes. And basically, it made no difference. So that's the sort of data you saw before, the correlated large dots. We also used small dots because we wondered if that would make a difference given their poor resolution. Didn't really seem to. And you can see for anti-correlated, for uncorrelated, really exactly the same. There's no difference between these conditions, which at the time I found really surprising because I had this idea from primates that, correlation was all about where it was. It was also kind of amusing, especially because there's all this literature about how humans have got absolutely fantastic, amazing stereo vision, and insects are incredibly crude and poor and probably doesn't even count as stereopsis at all. And I'm like, yes, and yet in this demanding stereoscopic experiment, the mantis can do the task just fine, and the undergraduates are a chance. So that amused me. So this was, of course, just a replication of the human experiments with the swirling disc, but it was no surprise to us that the humans couldn't do it. However, once we thought about it a bit more, I realised that actually it doesn't have to be so very different, and you can actually explain this result with something as simple as a high-pass temporal filter at the front end. So this little video here shows what's called a second-order motion stimulus. We also looked at these. So the point about this is that you see something moving from left to right across the video, right? But no actual dots are moving across the screen. There is no object that's moving. What's actually happening is that we have a notional target region, and as the target region passes over a white dot, that dot turns black and then goes back to white when the target's left it, and vice versa, the black dots turn white. And the mantis stereopsis works just fine with this second order motion as well. Now, if you imagine passing that through a high-pass temporal filter, that basically picks out where in this image are things changing. So, of course, all the background with all its complex depth structure just appears black because nothing's changing. And you just pick out the edges of the disk here, the leading and trailing edges, as that passes over. And once you've done that, now the problem is, okay, extract the disparity of the changing region, and that you could do with cross-correlation. So it's a really different kind of behaviour, but you realise that the underlying computations could be a lot more similar than we think due to a different, initial monocular processing. And so that's my kind of current understanding, as it says, of mantis stereo vision. Interestingly, since our work, I'm really happy to say,
but that you don't need stereo, right?
Oh, you don't need stereo to get where the disk is, but to extract the disparity of the disk you do.
Okay.
Do you see what I mean? Because we've shown that they can discriminate between this condition where it's crossed and this condition where it's uncrossed. So for that, you do need the disparity between the disk locations in each eye. Yeah. And that's what qualifies it to be called stereo. So Trevor Wardle, who was at that time in Cambridge, and I think Minnesota, used our 3D filters on his cuttlefish to investigate their stereoscopic vision. So it's another predatory invertebrate. And in their Science Advances paper, they share some fantastic videos in their supplementary material. Which this is what. So here's a cuttlefish in 3D glasses, and it's being shown a simulated shrimp on a screen, and it's attacking it with its tentacles. There's another example. There it goes into strike. So super cool. And again, they find really convincing evidence of stereopsis. So this is a key figure from their paper. And what they're showing is that as they vary the stimulus disparity, the position that the cuttlefish chooses to be in to strike at varies, right? Because they're always trying to get one tentacle length away from the object. And the distance that is from the screen depends on the disparity, implying that where they're perceiving the object isn't at the screen depth, but either in front of or behind the screen, depending on the disparity. And they also use correlated and anti-correlated stimuli, and they find basically no difference, so that the effective disparity on shifting the distance is exactly the same, whether the stimulus is correlated or anti-correlated. So intriguingly, even though, of course, cephalopods and insects are completely different invertebrates that evolved independently for millions and millions of years, it looks like they've both discovered this form of temporal change, stereopsis.
If they use first correlation, sorry, so you show that they mantis also is catch for uncorrelated stuff.
Well, it did in our experiment. So we showed the uncorrelated stimuli, and the mantis is striking when we arrange the disparity of the uncorrelated patches that are moving, such that it appears 2.5 centimetres in front of the animal. They do strike at it. I'm sorry, this is distance from the screen. So it says 7.5 distance from the screen, which is 2.5 from the animal. So they're striking at it when the disparity says it's in range. But when you've just got two discs going around the screen, but the disparity is wrong, then they don't strike at it. So for me, I can't think of another explanation other than, they're using the stereoscopic information. Is that?
Yeah, but that uncorrelated dots don't have, so they cannot use correlation, cross correlation as a.
They can't use a cross correlation of the luminance pattern. Yeah, absolutely, yeah. And I don't think they do. But they can use cross correlation. Imagine if instead of the dot pattern, I gave them the outline of two discs moving around the screen, right? Of course they could use the correlation there. And what I'm saying is I think effectively that is what I'm giving them, because they just see the leading and the trailing edges. And so the dots are just providing a texture of things changing as the discs move around. I think everything we have is consistent with that explanation at the moment. So that's my sort of working hypothesis.
Oh, good question.
Oh, great question. How do I see the rest of it? There we go. Rather than just for, yes, that's a really great question, Corey. Thanks. So are humans using both mechanisms? So there's some really interesting work by David Lee, where he tries to set up situations where this, he calls it kinetic stereo cue, is the only one available. And humans do show some very weak ability to use it. So yes, they may, but the fact that humans are massively impaired and the mantids aren't, means that for humans it can't be the only cue they're using. So, which is what you're saying, right? That we have the sort of complicated luminance pattern cross-correlation thing, and possibly this cruder temporal form, but the mantids only have the temporal form. Absolutely, I think that's a really interesting suggestion. One thing I think about with humans is we definitely have at least two forms of stereopsis, and the way I think about it is a little bit ventral and dorsal stream thing. We have the fine stereopsis, which is kind of what I've been talking about, and seeing discs in random dot patterns and fine depth structure, but you have to get your eyes to verge, because obviously we've got mobile eyes, so that's a huge problem. So I think we clearly also have another stereo system that's all about triggering vergence mechanisms. And I think there's a lot of evidence that that may be, it may use very different mechanisms and may sense forms of disparity that we may not even be perceptually aware of, but perhaps it does use this kind of temporal kinetic stereo. So absolutely, I think that's something very much worth investigating. Okay, so just skipping ahead there. Yes, so this is my summary of where I've got to so far. I think science has been pretty slow to appreciate stereopsis. We've misunderstood our own binocular vision, we haven't recognised disparity, and for obvious reasons, we still tend to think about it in a very anthropocentric way, and I definitely count myself in that. I've really had to sort of rewire my thinking about stereopsis in order to understand these insects, which is why it's been so fun, I think. Whereas the sort of fine, anyway, cyclopean vertebrate stereopsis does all this cool stuff like breaking camouflage and mapping depth structure, it's complex and computationally costly. So the invertebrate stereopsis works on temporal change, and something else I haven't really talked about is I think it's only designed to work on isolated targets. So I think it breaks down in very complex scenes where like everything's changing all at once. But that's okay, because they are only trying to catch isolated prey items. That is clearly still useful to them, and I'm interested in exploring robotic applications where it could be useful. And clearly it's not going to be useful for all applications. The key is going to be finding the right ecological niche. Now, I think I'm about 40 minutes in, about 10:45, so I'm going to, if you're okay with this, I propose to talk a bit about the physiology that we've been doing, to try to look inside the insect brain and see what goes on. And one of the attractions of working on insects is the thought that it's potentially easier to get to grips with that, just because their brains are so much smaller and different techniques exist. And this work is all done by Dr. Ronnie Rosner while he was in my lab. And again, I've got a video. It's a sad thing the sound isn't playing for this, but I'll just show it because it's nice to see the lab setup. So this is Ronnie's lab, and the chap there is actually asking, what's that sound? Because you can hear the crackling sound, and Ron is explaining that is action potential spikes in a visual neuron that you could see on the oscilloscope there. There's the mantis, immobilised once again in plasticine, and what Ronnie's done is expose the back of the brain by cutting away the cuticle. The mantis is face down, viewing through these coloured filters again, and we're displaying that swirling disc that you saw, and Ron is trying to find neurons that respond to it. So first of all, he tries to find any visual neuron, and then he, if he's lucky, he'll find a neuron that responds to that stimulus. Now he's using intracellular recordings, so he's then, when he's finished an experiment, he fills the neuron and then takes it to the imaging centre, and he's able to completely reconstruct the entire neuron and find out exactly where it connects to in the brain, which is really fantastic. So he's showing there one of our columnar commissural neurons, which, as you can see, goes right across the mantis brain and connecting the optic lobes, the visual areas on the two sides. And we think that may be a key neuron, as I'm going to talk about in a moment. Okay, so yeah, Ron is saying there, we think those neurons are active when the mantis is launching this stereosensitive strike. Okay, so in that way, Ron has been able to identify distinct classes of disparity-tuned neurons. He knows they're distinct because they look very different and they connect different areas of the brain, and he also has the physiology. And I think that's really cool because it was the first disparity-tuned neurons found in any invertebrate, and Ronnie worked so hard for this because the preparation is incredibly difficult and the recording is very difficult, and honestly he's got about sort of 24 neurons in total, I think, for years of work. It's really, really hard, but I think worth it. I've got, again, you're going to hear the crackling spikes here, but you can see on the screen the spikes elicited from one of his neurons. He dubbed it Tau Pro based on its anatomy, and that was the spiralling disc simulated to be at 2.5 centimetres, and you can see we've got a lot of spikes. And then this is with the disparity swapped over, so it's now the lines of sight diverging, and you can see the response pattern is completely different, and we're getting fewer spikes. So just summarising that information, at the top, the little sinusoids show the X and Y position of the disc as it spirals around. The raster plot shows the spikes recorded and successive plots. I'm afraid the control condition's now on the left. And the bottom shows the firing rate when you average over all those. You can see this much stronger response for the near disc. So that was really exciting. When we got a neuron like this, it was definitely cause for celebration. But I'm aware that you don't even know that this neuron is binocular, right? Imagine it has a receptive field that happens to be more activated by the crossed disparity than the uncrossed. It could even be a monocular neuron. So to really try and get access to what's going on, we used a much more controlled stimulus, which we call the disparate bar. And here's an example shown here. So we've got green and blue bars on a cyan blue-green background. And then the idea is that seen through the filter, that appears to the mantis as a black bar, but in different locations in the two eyes. Now, you have to remember, Ron's experiments are really demanding and really difficult. And when he gets a neuron, we have no idea how long he's going to be able to hold it, but it's not going to be long. And everything's also noisy, so we want lots of reps. So which means that the individual stimulus has to be very quick to run through. So we use only 6 different bar positions. Luckily, mantids like big prey. They typically strike best for prey that's about 10 degrees in diameter. So we use 13 degrees for the diameter of these bars. We made them vertical so we didn't have to worry about narrowing down to find their vertical location. And of course, we wanted to vary the horizontal position to look at the disparity. So we have 6 different positions, and then we present pairs of these bars. So when you've got a pair of bars, it corresponds to a virtual bar shown by this little black thing here in space, depending on the position of the bars in left and right eyes. So when I show you the results, I'm going to present little matrices like this, which have the position of the bar in left and right eye. So, and the pseudo colour is going to represent the neural response. So here I'm plotting this at column 3, because the bar's at position 3 in the right eye, and row 4, because it's at position 4 in the left eye, okay? And we're going to do this for all possible bar positions. We have 36 in total, right? Now, some of those represent virtual bars, as they would be in space. Other locations don't. So this point below the diagonal, at column 4, row 3, doesn't, right, because the lines are sighted divergent. So we tried to choose a situation where we've got 21 bar combinations that correspond to locations in 3D space. 6 are on the screen, and the remaining 15 are in front of the screen. And you'll notice we've chosen situations so that some of those combinations are very close to the catch range, so this 26 millimeters. And then we have 15 kind of control situations, these orange under the diagonal here, that I can't plot because they don't correspond to locations in space. And that's just zooming in a bit on that region close to the mantis. Okay, so that tau pro neuron, and you saw its response to the swirling disc. Here's how it responds to those bars. And the marginal plots, they aren't sums or anything. Those are the response to monocular bars presented in each eye. So you can see this neuron doesn't respond at all to a monocular bar in the right eye, does respond to a bar in the left eye, but when you put bars in both eyes, it is sensitive to the disparity between them. I'm just going to upsample to basically make it look nicer. Now let's mark on the region of peak response, and now let's map that back into space. And you can see it's pretty close to where the mantis is going to be catching its prey. So that's obviously exciting. This neuron is interesting because it receives input in the lobular complex in the optic lobe, but in just one side of the brain. It projects to a region of central brain, suggestively where the descending motor neurons receive input. So although we don't know, it's certainly got the right anatomy to be sending a strike now signal down to the ventral cord. So I'm going to,
Since this was a seemingly monocular neuron, or is this matching up the anatomy?
So, introducing my next slide, I'm going to represent this sort of schematically here with these little cups for input and the little arrows for output. So yeah, isn't that interesting? It's unilateral, it's on one side of the brain, but it's already binocular. So that's intriguing. And in fact, if you look at the response, you can deduce that it's receiving excitatory input from the left eye, okay? But it's receiving inhibitory input from the right eye. So the strong response to a monocular bar in the left eye that we see in isolation when it's just presented on its own can be suppressed by a bar presented in the right eye at the edges of the screen. So inhibitory input from the right eye. In fact, yeah.
So are these stationary bars or are they flashing?
They're flashed.
They're flashed.
That's right.
So the flashing is important because of the temporal change.
Exactly. Of course, it's also important for us because we're running through the experiment quickly. But yeah, it means that the temporal change suggestion, it will be picked up.
And so do you infer that this neuron then is downstream of whatever that temporal filter is?
Yeah, I mean, that would be my guess.
Okay. If you just put a bar on, does it not respond to that?
I think kind of like primary coordinates, when you put it on, you'll get an initial transient and then it will just die away as the bar sits there.
Yeah, okay.
Okay, so it's getting binocular input, which raises the question, where is it getting it from? And the answer may be the next neuron, COCOM standing for columnar commissural neuron. That's the one you saw in the video that went right the way across the brain, which is this is zooming in on its inputs and outputs there. So interestingly, it's homologous to a neuron called LC14 in Drosophila, and so potentially we might learn more about it from the Drosophila people. I'm actually going up to speak with one of them tomorrow, so that'll be good. It links the outer lobes of the lobular complex. It receives input in the optic lobe that's on the same side as its cell body, and it sends output to the opposite optic lobe and also to the central brain. And it too is disparity-tuned and selective to distances in the catch range. So here's its response, or the response of an example COCOM neuron to the spinal disc, and then in this sort of binocular matrix that you saw for the tau pro neuron. So again, we have a very sort of precise response to disparity in this critical catch range region.
Jenny, I see clearly the spatial change. Can you give me an idea of how long it takes for those changes to occur?
Sorry, Bob, which changes?
The positional changes or changing over time, and about what is that time? I mean, is it seconds or minutes?
So you're basically asking about the time constant, the filtering.
Yes.
Yeah. I don't have a great number for that. I don't think we tried to fit it exactly, but it's like 50 milliseconds. It's definitely not seconds. So you flash on a bar and off again, and you get this brief response.
Oh, better than we are. Probably.
I don't know. I don't necessarily think so. So these stimuli, the bars are flashed. The bars themselves are not moving, but so they're flashed at a position, pair of bars, and then we flash another pair of bars, and then we flash another pair of bars, and then these plots are showing the average response in a window, shortly after the onset of the bar. So we do get a response to these flash bars. We've not really gone after, what is the precise time constant of that response, how rapidly, does it decay if you were to have a sustained bar, mainly because we've been able to do very little experiments on these neurons. We haven't held them for very long. But I certainly regard them as consistent with a high-pass filter, with a time constant of 50, 100 milliseconds, which absolutely we would, that's consistent with our own kind of vision.
Okay, thanks.
Jenny?
Yeah. Two questions.
Yes, I can't see the room, so I'll rely on you to direct me. But just a quick methodological question. If I didn't catch it, do you paralyze the animal so that you can keep the recording stable? So, it doesn't strike and then destroy the the electrode placement?
Very much.
And then I guess the related question is, if not, do you have any sense that if you do paralyze the animal, if you affect the neural tuning?
So the animal isn't paralyzed, but pharmacologically, it's just immobilized. So again, Ronnie, we'll put it in the freezer briefly to kind of effectively paralyze it, pin it down with plasticine, so it can't move and it can't strike, which is very critical because these recordings are so delicate. He's trying to get a sharp electrode into this fibre, a few nanometers thick. So, and it's all on an air table and everything. So we're trying to keep it as immobilized as possible. He even tied off the gut so that the pulsating movements of the gut would not cause the brain to oscillate. So, but apart from that, as far as we can tell, all the usual neural machinery is carrying on. So quite probably a signal is being sent to strike. It's just that we don't see the strike because the legs are immobilized. And you talk about, wouldn't it be cool if you could try and record a motor signal, from its coccyx is trying to strike, and we just never managed to add in that complexity.
Yeah, it'd also be interesting to know if you do paralyze those motor neurons, if you somehow impair the neural, like if there's some sort of feedback that the creature's using, you might get access to that mechanism.
Yeah, absolutely.
No, super cool. Anyway, thank you.
Thank you.
Jenny, I should have thought of this before. We can roll the podium so that people can actually see you on the camera.
Then I've got my back to you guys.
Just a little bit.
Just a question. In what condition when we have stimulation? we still have a responsive neuron.
Yes.
When the stimulus is in a certain position in space. How you...
Well, I think about it as having a receptive field. So we have actually modelled this neuron. I think I haven't put slides on that because I knew I was going to be out of time, but it can be explained quite well by having a centre surround receptive field, excitatory centre, inhibitory surround, and it's set up so that the disparity between the excitatory centres matches the preferred disparity of the cell. So if you have a disc going over that receptor field as it passes around, I think this is more or less what you would expect, these little bursts of activity.
It's a monocular response.
Yeah, I mean, you see this neuron does respond to a monocular bar, it just doesn't respond as much as if you also have a bar in the other eye, which is why I say it's disparity-tuned. But, in fact, behaviourally, you can model the behaviour of the mantis. We've done this as well really nicely with just one such neuron. You just have to add a really expansive nonlinearity to basically make sure you only pick up the very top of this response, so you don't get responses to the suboptimal stimuli. And who knows, maybe that's indeed what's going on.
Oh,
Yeah,
so
thank you.
I just noticed that these are same neurons, right? Different trial, the rest plus.
Yes, all plots on this slide relate to the same individual neuron.
It's like they behave oppositely. For example, try 1 and 5 and the rest of them. Especially if it's like a tonic and then a little bit of inhibition.
That's a really good point. So you mean like why the raster plot labeled 5 and 6 has got so many more spikes?
Not only many spikes, it actually like a reduced spiking during the last sort of a spark. I mean, seemingly.
Maybe. I guess I'm seeing the region of inhibition staying fairly constant across the rasters, but I agree the background spike rate is going up a lot. And I'll have to ask Ronnie about this. One thing that may be going on here is he may have started his injection. So when he's filling the cell, he's using the current to drive the stain into it. So it's just possible he had started that on the last repetition, and that's why the spike rate suddenly gone up a whole lot.
How many of these neurons are there? This type of neuron?
And we don't know. We know there's more than one. They're very narrow in where they ramify, and it seems like there's a whole bunch of them covering different regions of space.
If you inject current into them, can you elicit strikes? Can you declare?
Again, we don't know, because you would have to be recording the motor neuron activity, and we just haven't done that. I know it's like, I think frustrating. But the trouble is, you can't just go in and say, let's record from a COCOM neuron.
Yeah.
So, I mean, Ronnie was doing this day after day for years and got like 4 of them. So,
Oh wow.
yeah, and that hence his reluctance, I guess, to be like, oh, why don't you now try feeding off the motor neuron activity as well? You know, to like don't complicate things.
Right.
Yeah. But it's fascinating. It's so tantalising. Yeah. Anyway, so given that arrangement, the receive input in one lobe and project it to the other, that suggested to us that pairs of COCOM neurons might be suitable for combining binocular information. So as shown here, you might get this reciprocal relationship where this neuron's picking up information in one lobe, directing it to the other, where it can then get picked up by its partner neuron, and now they both end up with access to binocular information. So that's speculation, but that's something we hope we might be able to get an insight to by, for example, tracing the connections of LCE14 neurons in Drosophila, where that kind of connectivity is potentially could be extracted. Just to show you a couple other examples from our mantis neuron zoo. I know I'm nearly finished because I know it's past 11. We found this interesting neuron TME-SEN, and that intrigues me because it has its input sites in the central brain and its outputs in the medulla, which is one of the earliest visual areas in the optic lobe. And I'm just showing the example disparity plots for 2 TME-SEN neurons. Not quite as beautiful as the others, as you'll see, but still disparity-tuned. So what's that neuron doing? You can see it's really sending a lot of information. Each one of these beads is a synapse. You see it's all the way across. Of course, I don't know what it's doing, but I wanted to speculate that it might be involved in visual attention, perhaps, guiding that there's something nearby. Another information that neuron that sends feedback is what's called tar-sen, and that was intriguing because these neurons systematically responded better to bright than dark features, which is unusual for mantis. And they have super large receptor fields, and their binocular maps are, because they're kind of the wrong way around, right? So now we've got the biggest response below the diagonal to stimuli that aren't even within range. So what are they doing? It reminded me a bit of the tuned inhibitory neurons in primary visual cortex that certainly Enrico will be familiar with, and maybe some of you, which seem to be doing the wrong thing. They respond more to anti-correlated stereograms than correlated ones. And so if you look at this neuron, I'm trying to back project, where is it looking for? It's responding to bars in the right eye at this sort of location, and in the left eye at that sort of location. So it's suggestive that mantis prey are usually dark objects against a bright background, and this neuron almost seems to be looking out for the background rather than the bug. Could that help it inhibit strikes to excessively large objects? I don't know, but that's the sort of thing I'm thinking about. And so putting that together in this sort of diagrammatic form, here is some of the circuitry that we've uncovered about mantis stereopsis. And because I couldn't resist putting together a bit of a working hypothesis about what this might be doing, it's interesting we've got these feedback loops, which I don't think we've got proof of in primate stereopsis, although we certainly believe they exist. So maybe, this is my here's one working hypothesis to shoot down over the next few years. CoCom neurons do the initial binocular combination, as suggested, and then the central brain gets some information about that from CoCom. And then it can give that information to TME-sen, and maybe those are helping guide the spatial attention. Maybe tar-sen neurons are just checking, okay, what have we got here? Does it really look like prey? Is it small against a bright background? Feed that back to early visual areas, again, perhaps for guiding attention. And then Tau Pro can pick up that information from the early visual areas at the end of the feedback loop and potentially initiate a strike. Now that's all speculation. But it's intriguing because I think what I take from this is just how complex the circuitry was. When we started out, one sort of live hypothesis about mantis vision is it's all monocular processing. One side of the brain finds a prey item in the left eye, other in the right eye, and then there's basically an AND gate at the motor neuron that launches the strike. And it's very far from the case. We've got this surprisingly complex circuitry with many classes of disparity-tuned neuron and this interesting multiple feedback loops. So I'd love to find out what it does, and I sort of have hope that just maybe in insect brains it might be easier than in primates, just because they're so much simpler and more techniques are available. Well, I'm sorry I'm well over time, so I will leave it there, and thank you very much for your attention. Great, any other questions?
Have you tried more like just a sort of labeling technique, because it might be more amenable to more relevance of the cell, like intracellular?
So a different according to technique than intracellular?
Yes, because you might have more the spike activity, and then just with one extracellular, they label the cell, you might get more cells.
So yeah, I should say I'm not an expert in these techniques. Right? I was the PI, I've been very supportive of making tea, and Rod is the postdoc doing the actual experiments. My understanding is if you want to label the cell, you have to pierce it, so you have to use the sharp electrode. And it's not the labeling that causes you to lose it so much. It's just that you always have to make this decision, because once Ronnie can only stain one neuron, basically, because if he stains two, he won't know which is which. So it's like, should I carry on recording from this one, or should I stain it now? And sometimes you're like, I'll just carry on recording a bit, and then you've lost it, and now you can't stain it. But if you do stain it, then potentially you could have done more before you started injecting the current and changed its behaviour. So if you want to know the anatomy, I think you kind of have to do that. If you're happy not to have the anatomy, you could do extracellular recording. And coming from monkey brains, I was much more relaxed about that. I remember having this conversation with Ronnie early on, and I'm like, why don't you do extracellular recording? It'll be so much easier. You'll get the spikes, but you won't have to hold the cell. Ronnie's just like, he was from an insect tradition. He said, but then I won't know what the neuron is or where it connects to. And I'm like, welcome to my world. But he was very much wanted to go after having the anatomy as well. We are trying, or interested in developing neuroimaging techniques, so like calcium imaging of the mantis brain, where again we won't get the beautiful anatomical information, but potentially it'd be much easier to find out what's happening at a kind of like MRI level of resolution in the mantis brain. And I am talking to colleagues and trying to get the collaboration. We haven't done it yet. I see I've got a question in chat. Thanks, Bob.
So one question is, how much variability do you think there is? So since you have a few, I mean, you have 24 neurons, you said, and how much, so do you think all of these 24 would be identifiable in any mantis? Or is it, in insects, it seems like there's quite a little
I mean, exactly, that would be my guess, because it is a world where you can say, you know, VLC14 neuron in Drosophila. So yeah, our assumption is that these neurons are found basically in every mantis. I don't know how much variation there is, like, you know, do they all have the exact same number of co-com neurons, or is there some variation? I don't know. But yeah, certainly that's our thinking. And actually, so one small complication is that the behavioural work was done in the African lined mantis, Photomantis lineola, which is basically the commonest hobby mantis in the UK, and so the cheapest and easiest to get hold of and super robust and so on. But it's a little bit smaller than the ones Ronnie used. So Ronnie, for obvious reasons, wanted the one with the biggest brain. So he has done the neurophys work, is in Hierodula, and Monodera as well. But we didn't find even differences between those genera. I mean, of course, I'm sure there are differences, and if you could drill down in more detail, you would find them. But yeah, we're thinking it's the least carrier.
So Jenny, you mentioned the A word, attention.
Yes.
In association with the feedback signals.
Yeah.
And I'm curious about how you're thinking about it. So one circumstance in which I could see that feedback might be useful for the praying mantis is that situation where you said where something enters the capture zone and then is motionless,
right, and
it doesn't strike, but it seems sort of poised to strike, so that if it makes any little mistake in terms of moving, then the mantis can. And so I'm curious if it would be something like basically implementing a prior, that there could be something at that depth that cannot be yet detected in that moment visually, but they have a prior basically about it being there.
That's exactly it. I'm not sure I'd use the word prior, but exactly.
It's loaded, but sure.
Yeah, exactly. But that's exactly what I'm thinking. Okay. So if you put these two halves of the talk together, if it's got a high pass temporal filter at the front end, then when the insect's frozen and the mantis is still, there's no signal, right? So as I say, but they're still very much aware there's something there. And you sometimes see them do these peering movements, which naturally people think of motion parallax, they're trying to get depth information. Possible, and they do use motion parallax in that way, we know, for jumping and so on. But my guess is that they're simply trying to turn their vision on. They're like the T-Rex in Jurassic Park, you know, if you freeze they can't see you, yeah, but if it moves, it generates retinal slip. And so maybe this feedback is saying there is something at this location in space, you know, keep looking at that, you know, maybe. Got a question, Dan, hey Dan.
Yeah, hi. Yeah, this is totally lovely. I don't want to distract from some of the main points you're making, but there's this thing in the back of my mind that I could ask you about the whole time, which is that there's this, maybe in primate, maybe not in insect, but there's a second reason to have two eyes, or maybe a problem, and even depth deception, at least naively, which is, you know, you have two sensors facing the world, and there could be occlusions and other things that, and a problem of divergence and disparity detection is simply to just make sure you can use both of those sensors when they're relevant and not. And how much would that be a viable second functional hypothesis of combining the inputs for two eyes? And would it apply to the insects as well as humans? Or is it sort of a little naive?
I think that's a great question. And for insects, it almost applies more than for humans. So I read this review recently on binocular vision across the animal kingdom. And one of the things I was thinking of with insects, because they've got these compound eyes, you have the possibility of viewing the entire world all around you from a single optic center. Do you see what I mean? So physically, it's two eyes on either side of your head, but you've just got the entire optic array. Right? So you could do that, and then you wouldn't have this problem of matching up, because you've only got one line of sight from this optic center, and that would be so simple. So why are they not doing it? Right? Why does even Drosophila have a kind of two-ommatidial overlap? it seems like it really complicates things. And so one possible answer is, for example, improved contrast summation. So if you're a nocturnal bee, which apparently exists, maybe that's a good reason to have two lines of sight to the same point in space. But if you're not a nocturnal bee, that seems like a pretty marginal benefit. And then you start to think about, well, what about stereopsis? So I agree, it's sort of like if you're going to have a binocular overlap, it's sort of, you might as well have stereopsis. But I think certainly for insects, the bigger question is why evolve a binocular overlap?
Yeah, thanks.
So since you also work on clinical and industry applications, do you see any translation of the findings of magnets to the clinical models?
Yeah, great question. Yes, I do, and that's one of the things that I find fun about working in these different areas. Even though it means I'm spread a bit thin, I really enjoy the sort of cross-fertilisation between them. So yeah, I'm interested in the applications for robotics of this form of mantis stereopsis, and I'm trying to collaborate or develop collaborations with roboticists to explore what kind of performance may be possible in particular applications. I've got a whole separate talk about mantis stereopsis without correspondence, but basically I'd like to explore how well it could work in robots, because I think it's such, on the face of it, it seems a really stupid way of doing stereo. I think if you just presented it to an engineer and said, hey, why don't we design an algorithm like this, people would be like, oh, that's dumb. But if you can say, well, this insect has been doing it for millions of years, maybe it's not so dumb after all, it really motivates you to go after the benefits of that algorithm. So that's one application. Clinically, it's perhaps a bit more of a stretch, I absolutely agree, but we touched on this earlier with the question about vergence, so what are the mechanisms that bring the eyes into alignment, and have we evolved some of the same mechanisms that apparently mantis and cuttlefish have evolved, and therefore would it be helpful to bring that way of thinking about it to studies of human vergence control that might be relevant in the development of business. Well, thank you so much for being an awesome audience. Cheers, everyone. Thank you.
Oh yeah, well, for people who are here, we are going for lunch soon. So if anybody wants to join us, please feel free to do so.